all. This is Dr. Mubin Sayed from drbeen.com. Welcome to one more show. So the discussion today is a new preprint study by Dr. Bruce Patterson and Dr. Ram Yugandhra and their team members. The study says that in the vaccinated individuals, the components of spike protein S1, mutant S1, I'll explain later, and S2 can be seen up to 245 days after the vaccination in the individuals who are suffering from post-acute sequelae of COVID-19-like syndrome or long COVID-like syndrome. This happens to all 4 types of vaccinations, that is AstraZeneca, Oxford AstraZeneca, Moderna, Pfizer BioNTech, and Johnson & Johnson. This is a significant study in the terms that in this study, the S2 component of the spike protein and mutant S1 component of the spike protein were not observable in the long COVID patient and are seen in the vaccinated individuals. This is a big deal. So vaccinated individuals who have long COVID-like symptoms have even more proteins in them made up of spike species compared to long COVID patients. So this is the study. They have a potential management proposal as well, how to manage it, and that is CCR5 antagonists and statins. Again, this is not a medical advice from me. This is what is in the study, so you can look at that paper as well and read it by yourself. So let's very quickly look at this study together. And I'm going to turn off my camera for a few just so that we can look at the whole screen. So this is a study. Persistence of S1 spike protein in CD16 positive monocytes up to 245 days in SARS-CoV-2 negative post-COVID-19 vaccination individuals with post-acute sequelae of COVID-19 like syndrome. So this is a study, interesting study. I have gone over this, all of it. So we will discuss a little more. Let's start with my diagrams. So this study, you will notice as we go through this, is a lot about the monocytes. So here are the monocytes. Keep this in mind that this monocyte that you're seeing here is in 3 kind of states. This state has a lot of fire burning on it. This state has some fire and some water hoses as well. And this has a lot of water hoses. I'll explain that a little later. So this is a study, as we just saw, that Dr. Bruce K. Patterson, Dr. Ram Yugandhra, and then other team members. Now, I'm going to discuss 2 things up front. Number 1, the abstract, and number 2, the potential management. Then we'll go into the details of the study and discuss the mechanism that is responsible for the post-acute sequelae of COVID-19 like symptoms in the vaccinated individuals who have them. So here, there have been concerning reports about people experiencing new onset persistent complications greater than 30 days following approved SARS-CoV-2 vaccinations, Pfizer BioNTech, Moderna, Johnson & Johnson, and AstraZeneca. We sought to determine the immunologic abnormalities in these patients and to investigate whether the potential etiology was similar to post-acute sequelae of COVID-19, PASC, or long COVID. We studied 50 individuals who received one of the approved COVID-19 vaccine and who experienced new onset PASC-like symptoms, along with 45 individuals post-vaccination without symptoms as controls. We performed multiplex cytokine, chemokine profiling, these are the chemical substances released from the cells, with machine learning, as well as SARS-CoV-2 S1 protein detection on CD16 plus monocytes. So I'll explain that a little later, what these monocytes are, subsets using flow cytometry and mass spectrometry. We determined that post-vaccination individuals with PASC-like symptoms had similar symptoms to PASC patients. So the long COVID patients from the virus and the long COVID-like patients after the vaccine had similar symptoms. When analyzing their immune profile, post-vaccination individuals had statistically significant elevation of soluble CD40L, CCL5, IL6, IL8. Machine learning characterized these individuals as PASC using previously developed algorithm. So keep in mind IL6, IL8, CCL5, soluble CD40L. Of the S1 positive post-vaccination patients, we demonstrated by liquid chromatography mass spectrometry that these CD16 plus cells from post-vaccination patients from all 4 vaccine manufacturers contained S1, mutant S1, and S2 peptide sequences, protein sequences. Post-COVID vaccination individuals with PASC-like symptoms exhibit markers of platelet activation and pro-inflammatory cytokine production, which may be driven by the persistence of SARS-CoV-2 S1 proteins in intermediate and non-classical monocytes, and I'll explain those in a second as well. The data from the study also cannot make any inferences on epidemiology and prevalence for persistent post-COVID vaccine symptoms. So they're saying we cannot tell from this data that how prevalent the vaccine, post-vaccine long COVID-like symptoms, or what is the epidemiology of it. Thus, further studies and research need to be done to understand the risk factors, likelihood, and prevalence of these symptoms. So, friends, this is the only time when I read so much of the text, and for the future next slides, there will not be such lengthy wall of tests. Okay, so potential management first. We've now seen what the summary of the study is, potential management. And again, this is from the study. One part is mine and I have written my name on it. CCR5 antagonists. Because CCR5 is increased and then can lead to clotting, they are saying that the reduced monocyte recruitment to the site of atherosclerosis. So giving CCR5 antagonists will reduce monocytes to arrive in the area of the blood vascular endothelium and stick there and cause inflammation there. So that is one. Secondly, statins. Reduced fractal kine expression leading to reduced monocyte binding to endothelium, and I'll explain that in a second as well. Finally, Mubin's point of view. The mutant S1 that is only found in post-vaccinated long COVID-like patients is interesting. Mutant S1 was not found in post-virus long COVID patients. So maybe remember that study that we had a few days ago where this study demonstrated that in the vaccinated individuals, when the spike protein is being formed because of the mRNA synthetic changes to it, that causes ribosomal slippage, which in turn causes the truncated proteins or partial proteins or off-target proteins to be formed, or as they write it here, the researchers in this study, the mutant proteins. So the other study that I'm mentioning, they had a proposal for how to fix the vaccine code so that it does not produce mutant proteins. So I think that would also be an important thing to do. But this is my editorial, this is not in the current study. So what is the new evidence? What have they found? Here is what they found. So first of all, let's start from this side of the diagram. Here we are seeing three colors, blue and the reddish and the greenish. These are the three spike proteins. So every one of these threads is actually a spike protein. What happens is when the virus makes these spike proteins, then three of them come together and intertwine with each other and become a trimeric molecule, or we call it trimerization of the spike proteins. Every spike protein that is taking part in this trimer is actually able to bind with ACE2. Normally, any one of these get a chance to bind to ACE2, but three of them are wrapped around each other together. What they found was, in the vaccinated individuals, in the monocyte of these individuals, and we have to discuss a little more in the detail, in the monocytes of these individuals, they found, number one, spike proteins S1 part. This is what they had found in the long COVID patients as well. However, they also found S2 part as well in this group of patients. And they had not found S2 in the long COVID patients. So this is an additional protein that is found in the vaccinated individuals, monocytes. And then here, if you see this mutant kind of D-shaped, mal-shaped, incorrectly formed protein is the mutant S1, and they found mutant S1 as well in the monocytes of the vaccinated individuals who had long COVID-like symptoms, but they did not find this mutant S1 in long COVID patients. So it looks like vaccinated individuals who have long COVID-like symptoms actually have more kind of protein stuck in their monocytes. So that is one they found. And here is my editorialization. This is the study that was printed in Nature, and I had actually discussed it. This study said N1 methyl pseudouridylation of messenger RNA causes +1 ribosomal frame shifting. I did this in discussion in detail. I am actually surprised that there has not been a lot of traction of this study. This study actually necessitates for us all, especially those who are designing the mRNA vaccines, to look at the vaccine and correct the vaccine design so that the off-target protein, as this study said, or mutant proteins, as the current study that we are discussing, do not occur. I did not see any traction on this. Maybe the vaccine companies are working and they don't need to tell us. That may be the case, but this is, I believe, a groundbreaking and important study. Okay, so one set of problems that they found, or the proteins that they found, were S1, S2, and mutant S1. The second thing that they found, as I said before as well, they saw more IL-6, more IL-8, they saw IL-1, interferon gamma, tumor necrosis factor alpha to be increased as well. Now, interleukin-1 is released by monocytes and macrophages during the inflammation. Interferon gamma is released by natural killer cells, these are here, and then CD4 cells, these are these, and then CD8 cells, which is here, T cells. And then tumor necrosis alpha, macrophages, which are here, CD4 positive T cells, which are here. So you might say that, hey, these cells look better developed, and that is because I'm writing a book on immunology and inflammation, and in that book I've already created some cells and their diagrams, and these are these. There are some cells whose diagrams I have not finished yet, and that is why you see these cells to be less rendered, working on them. But anyways, interleukin 6, interleukin 8, interleukin 1, CCR5, interferon gamma, tumor necrosis factor alpha. Now the question is, what is the potential mechanism of these symptoms? So again, this is a potential mechanism because there is a hypothetical conjecture here, and it's not the final mechanism. Although they saw the same, they thought the same things with the long COVID patients as well. I would say that I do not endorse or not endorse. I think I like some of the discussion. And then there are patients who have reached out and they have benefited tremendously from this protocol of CCR5 antagonists and statins. And there are patients who have said, we have not found any benefit. And there are patients who even said that we felt more aggravated after this. So is this the final pathophysiology in every patient? That may not be the case, but let's see within the context of the study what are we looking at. So here, first of all, trying to understand the three kinds of monocytes, and I'm going to hide my screen again so you can see the full screen. So here, first of all, monocytes can live in three states. A classical monocyte state, this is the state in which they were discovered the most often. That is why we call it classical. A non-classical state, which was not something that we were expecting, and then an intermediate state. Think of intermediate state as a state in which a classical monocyte, when it wants, it can convert itself to or the environment and environmental factors can convert it into an intermediate monocyte. Intermediate monocyte then can become classical again or can switch to non-classical. Similarly, non-classical can become intermediate and then become non-classical again or become classical depending upon the needs and the environment around it. So intermediate is kind of a neutral place from where monocyte pool can move towards inflammatory or non-inflammatory or classical or non-classical monocytes. Now the other thing that you would see here, that in the classical monocyte I have made these flame-like structures on it. These are the CD14 molecules. CD14 molecules are positive in classical monocytes. And CD16, which you see here on the extreme right side, these little pipes over here, because this is a non-inflammatory monocyte, I gave it these little cool water hoses. So anyways, these are CD16. So if you see here on the left side now, classical monocyte has CD14 molecules, a lot of them, but does not have any CD16 on it. Plus, if you see here, there is this little one arm present here, but just one or two or fewer CX3CR1 molecules. These CX3CR1 molecule are really important to keep in mind because this is the molecule that helps the monocyte bind with our endothelium. Endothelium is the cell on the internal surface of the blood vessels. This is the core of the discussion today, so we must keep that in mind. So if you see here, classical monocyte, or we also call it pro-inflammatory monocyte, has no CD16 on it and has CX3CR1 molecule as well, but fewer in number. Then if you see the intermediate, look at the shape of it or the molecules on it. There are some flame on it, but the flames have reduced, so that means CD14 have reduced in number. Plus there are some water hoses that have appeared, so that means some CD16 are now appearing. So you would say correctly that the intermediate monocyte is CD14 positive and CD16 positive. And then if you see these arms, the grabby arms for endothelium, these arms actually connect with the fractalkenes, which are molecules dangling on the surface of endothelium. Think about little bush-like things that are on top of the endothelial surface. So these arms can bind with them. These are CX3CR1, and you see that these are a little more on the intermediate cells. Finally, if you see here, non-classical monocyte, which are the hero of today's discussion, these are Of course, see, there is no flame structure on it, so these are CD14 negative, but there are lots of these water hoses on them, so these are CD16 positive. And if you look at the study, that's what they keep saying, that we found CD16 positive monocytes containing these proteins in them. Plus, if you see these grabby or binding arms, CX3CR1s are more abundantly present on the non-classical monocyte. So I could say over here that non-classical monocyte will more avidly bind with endothelium because they have more receptors to bind on the fractalkenes of the endothelium. Good. So today our discussion is going to be mostly with this cell. So now this is something that is going to be a t-shirt design as well. What happens is, this is a very important point today. A normal regular monocyte lives in the blood circulation for 3-4 days to 3 days. The monocyte from the blood circulation can move into the tissues and become resident there and become macrophages or dendritic cells. I know that immunologists over here would be saying that, hey, macrophages are not always derived from monocytes, and that is correct. There can be actually macrophages that would give rise to more macrophages and whose origin was not monocyte but macrophages are the cells that were present in the yolk sac of the fetus. So we're not going in the detail of how these cells, macrophages and dendritic cell, where do they come from. However, we know that the monocytes can migrate into the tissues and become macrophages. On the other hand, if they don't, they would just die in the circulation after 2-3 days. There is one more delicate point which is really important for us to catch, and that is, it is possible for the monocytes to enter the area of inflammation, for example, where the vaccine has been injected, start phagocytosing the antigen over there, for example, spike proteins, and then before they convert, or we will call it differentiate, before they differentiate into macrophages, these monocytes can hop back into the circulation and start circulating. These are the monocytes now carrying the spike protein in them and circulating. But of course, I'm going to now put on my picture here as well. Of course, you would say that, so what if they are in the circulation? Within 2 to 3 days, they are going to die with the spike protein in it. And I remember these discussions with Dr. Bruce Patterson when he would come on our channel and have this discussion. And in those days, we all used to kind of think a little more about how do these monocytes become immortal. Turns out, in this study, Dr. Bruce Patterson and team has mentioned that when the monocyte, so here is the monocyte, and this is an endothelial cell that is inside the blood vessel, the inner surface cell. When a monocyte binds with the fractalkene, so this blue little plant-like structure are fractalkene molecules that are dangling on the surface of the endothelium. Monocyte, as I had said before, has these CX3CR1 molecules, these binding arms, receptor arms. They can bind with the fractalkenes. When a monocyte binds with the fractalkene, that causes inside the monocyte a secondary messenger system to become activated, another molecular mechanism to become activated, which is called BCL2 mechanism. Here I kind of brought it out as this little wizard. So once again, let me take my picture out as this little wizard, which is really, this is the BCL2, and imagine that this is within the cell. And what BCL2 does is it imparts immortality to this monocyte. It is anti-apoptotic, meaning it would prevent this monocyte from dying. So of course the monocyte is going to say that, you know what, 2 or 3 days later I'm going to die and I'm going to do apoptosis, I'm going to kill myself. But if that monocyte was bound to the endothelium on the fractal gene, then the BCL2 will become activated and prevent the monocyte from dying. And now remember, we said that CXCR3 molecules were most abundant in which monocytes? In non-classical monocytes. So it is the non-classical monocytes that would get stuck on the endothelium and become activated and become longer living or immortal as well. So here is the t-shirt design that I'm going to put. So those who are maybe long COVID patients or long COVID-like symptom in post-vaccine patients can have this on their t-shirt that says, "My monocytes are immortal, ask me why." And the discussion can be about the BCA monocyte binding with the endothelium, fractal genes, which causes BCL2 to become active. BCL2 in turn makes the monocyte immortal, and now this immortal monocyte would continue to produce problems for the endothelium and cause activation of platelets and lots of other problems. Okay, so tell me this, do you like this idea of this shirt design? Okay, now we also know that in addition to the monocytes binding to fractal genes, here is an interesting and delicate point. Interleukin 1, interferon gamma, tumor necrosis factor alpha, all increase the expression of the fractal genes. So if I go back here, these little plants, I think that you can imagine that the more these plants are, easier for the monocytes to bind there. So now we have an interesting problem. There are chemokines and cytokines that increase the fractal genes. So that means that endothelium is easier to bind with. And then we have the non-classical monocyte with a lot of CXCR1 receptors that can bind with these fractal genes. They both are now increased, and so we have the perfect storm. Then they say in their study that stress and exercise increase non-classical monocyte activation as well. And the exercise can increase this activation by 4 times. And this would probably explain why people who have PASC-like symptoms after vaccine, of course they're stressed now, why they become aggravated or their symptoms, plus those who try to do exercises, why do they become more aggravated as well? And I have done another discussion about the muscle phenotype change in the post-vaccine PASC-like patient or long COVID-like patients and what happens to their muscles. I've done that discussion in detail as well. That is also worth looking at. Now, we have now proved that there is monocytes, non-classical monocytes, CD16, they are carrying these proteins, so they are active monocytes. They're going to get connected to the endothelium, and now what are they going to do? So here, this is the monocyte, non-classical monocyte, CD16 positive monocyte, connected with the endothelium. This is happening within the blood vessel wall. When it is connected there, it would cause inflammatory molecules to be released and it would cause local inflammation. When the blood vessel wall is inflamed, it would cause the factor 8 to be exposed. Now factor 8 has one Willebrand factor on it. These little dilatations are one Willebrand factors. One Willebrand factors that is now exposed in the inflamed blood vessel can bind with the platelets, or platelets can bind with it. That is actually the mechanism by which platelet clotting starts in a hurt blood vessel or injured blood vessel. So here the blood vessel originally didn't have an injury. It is a monocyte that came in and started that. So when the platelets in the vicinity, they come in contact with von Willebrand factor, these platelet becomes active. So when they become active, not only they would start aggregating with each other, but these platelets are going to start releasing the chemical substances. For example, these would release soluble CD40 ligand, which is a chemotactic molecule for neutrophils and monocytes. So in the area, more neutrophils and monocytes will be brought. Now please remember, this could be anywhere in this system, because monocytes here are not going to just say that, all right, I'm going to be stuck to the blood vessels of the arm. They're going to go and get stuck anywhere on the blood vessels. So wherever they are, they would cause platelet activation and bring in more monocytes and neutrophils. Talk about a perfect storm. Then these platelets would also release CCL5 and RANTES, and that would cause, or that would promote, inflammation. Finally, platelets would also, and endothelium, which is now inflamed, would also release VEGF. We have talked about this in the past as well. This is a molecule that would cause new blood vessel formation. Now you might say that, hey, new blood vessel formation is awesome, it is a great thing. But no, it is not a great thing. When we, in this kind of a context where the patient actually doesn't need more blood vessels, when you start making new blood vessels sprouting from the current blood vessels, the new blood vessels are, number one, going to be building. It's like building new blood, pipes, and these are yet leaky. They are not fully mature, and that would cause hyperpermeability or the congestion in the tissues, and that could be any tissue. And secondly, there would be a lot of blood vessels unnecessarily present in the tissue, which would cause tissue damage or tissue functional problems. So this is the potential mechanism. So once again, if we go back to the CCR5 antagonist for management and statins, what they do is CCR5 antagonists and the statins, they reduce the movement of monocytes, classical, non-classical monocytes, plus their binding to the fractalkenes. Result is monocytes will not arrive in the areas, plus they will not bind avidly to the cytokines, and the whole mechanism would become downregulated, and the patient would, in theory, start feeling better. So this is the discussion. Thank you very much for listening in. I would also recommend that you look at the actual study as well. Links will be in the description. Thank you very much. Please like, subscribe, and share, and I would see you the next time. Bye for now.